abstract, microscopic. It's understanding things that you don't actually deal with in real life. But your special senses, that is your connection to the world, right? How your eyes work, how your ears work, your smell, your taste. That is something you think about all of the time. So in total, the lecture is about senses in general. And we have two types of senses. We have the general senses, also called the somatic or visceral senses, depending on where they're located. So somatic senses is going to be in your skin, in your muscle. Visceral senses are going to be in your organs. So what do you think would be an example of a somatic sense? Touch. Touch on your hand, in your skin, anything like that would be a somatic sense. What do you think would be a visceral sense? Hunger. When you get a stomach ache and you feel that pain in your stomach. Heartburn. Yeah. The fact that you have to go to the bathroom, those would be visceral senses. And then there's the special senses. Smell, taste, sight or vision, hearing, and then balance. Okay? Usually, we don't include balance as a special sense. Our textbook does. It normally just kind of gets lumped in with hearing, because they're both coming from our ears. Just like with a reflex, the sensations are going to have a number of steps that have to be followed in order for the whole thing to go to completion. Number one, we have to have a stimulus. What do you think would be an example stimulus for a general sense? Touching a hot stove. We sense something hot. It is sensed by a sensory receptor. It's sensory. Remember, sensory things are going to sense something in the outside world. It has to then take that sense, what it detected, and turn it into a nerve impulse. And so when you detect something, that's going to open the first few sodium gates, which is going to bring it up to threshold, and when you hit threshold, it's going to fire, and that's going to be your nerve impulse. After that, that nerve impulse has to go all the way up to the brain. Your senses are going to be in the brain. So we're going to come up the arm, into the spinal cord, up the spinal cord, into the brain. And then once it gets to the brain, it's going to get transferred to different places in the brain. Different places in the brain are going to control and monitor the different senses. There's going to be a spot for sight. There's going to be a spot for hearing. There's going to be a sight for balance. They're all going to be in different places. So once it gets there, the brain is going to integrate that. Integration is a term we already learned. What was integration? Decision making. Yeah. It's going to take the information that it has coming in, and it's going to put it together to make one coherent idea, and then off of that idea, it's going to make a decision. And so, as an example, we'll touch the hot stove. What is the integration? What is the decision that the brain is making?
Remove your hand.
Actually, a step before that. So the sensation, it's hot. And so there was a sensor that the heat did something in my hand, which brought something up to stimulus, then it fired, went up to my brain, and the brain then integrated that sense and said, it is hot. The brain then made another integration to say, if it is hot, I need to pull my hand back. For a special sense, imagine I have the light coming into my eye. The light comes into my eye, hits the receptors on the back of my retina, which sends a nerve impulse to my brain, and in that case, it's not nearly as, it's not really a decision-making process. It is one point in the picture of the world around me. That is going to be the integration. It senses one beam of light of a certain wavelength, of a certain intensity, came from a certain direction, and so that is that pixel on my screen that I see the world with. If you take a whole bunch of those, put them together, then you get the whole picture. The ability to be aware of your surroundings, because of these senses, is perception. And so we're aware of it, and we know what it means. And so that is, yes, if I touch this, I feel that it is firm. I'm aware that there is something firm here, but I also then make the conclusion, and I interpret that to mean this is a solid. This is something that I can't walk through. Somebody say, I should try?
No, you can try.
Oh, I can try? Yeah, I can try anything. I can try to touch the ceiling. It ain't gonna happen.
You can jump. Good job.
I can jump, yes.
Shoes on, you got it.
Yeah. That's what MJ wears, right? Adaptation. Adaptation. If you do something, if you have a sensation for a long period of time, generally it's going to become weaker. What's an example of that? There's a lot of examples. What do you think is one? Say it again?
I was saying, like, pain.
Pain?
Like some chronic pains.
Yeah. With chronic pain, generally after, unfortunately, months and years of the same pain, your brain actually kind of starts to block it out.
Really?
That's true.
Because my mom still complains.
What's something else? Something hopefully
I
more
was going to
everyday.
say when you touch like cold water, it eventually isn't
Yeah.
that cold anymore.
You touch cold or hot water, it immediately feels hot or cold. But then after you leave it in there a little while, it doesn't feel as cold. But something else. Anybody have air fresheners in their house?
Oh, your smell
Oh,
goes away.
they go-
Yeah. The longer you smell something,
goes blind.
you don't smell it anymore. That's why generally, every time you walk into someone else's house,
You smell
it seems like their house smells. But in reality, yours does also. You just don't smell it.
You just don't know it.
People who have 15 cats, they say their house doesn't smell. Certainly it does. But they are nose blind to it. After a while, their mind blocks it out. Why do you think, if you live in a house with 15 cats and it smells horrible, why would your mind block that out?
Because it gets used to it.
It gets used to it, but there's a reason that it gets used to it and decides, I am not going to smell this anymore.
Because it's horrible.
You don't want to, there's too much stimulus, so you kind of selectively choose what to not pay attention to.
I think you're on the right track. So, if there's a room with one very, very strong smell, are you going to smell the weaker things?
No.
No. But if you block out that strong smell, now all of a sudden you can smell the weaker things again. And so it's kind of, when you first walk into the room, it's new, and so it just hits you. And so your mind is saying, holy cat crap, it smells really bad in here, okay? Maybe you need to turn and run because your sympathetic system is kicked in. But after a while, if you stay there, your brain says, okay, it must not actually be dangerous. Let me cut this smell out so that I can smell something else. These receptors that are going to lead to this come in three flavors. They're going to be free nerve endings. We can have encapsulated nerve endings, or there can be specialized cells. So imagine you are that person laying on the floor and they are beating on your chest. What sensation do you think you are going to feel?
Pain.
Pain. Okay? Pain is a sensation that is felt by free nerve endings. If you remember the diagram and the model of the integumentary system, the skin, remember there was some free nerve ending. It just was a nerve that just kind of ended, and it went nowhere. That is a free nerve ending. What's something that you think from that skin model that would be a receptor or a sensor that is a separate cell?
It's your touch and the pressure, but I don't
The
know.
touch and the pressure things. What were they?
Oh, yeah.
Meissner's and
The other one I can't
Pacinian, what's the second word?
Corpuscle.
Corpuscles. So those are actually individual cells. They're very specialized cells. Encapsulated nerve endings are essentially, if you can picture the free nerve endings, they kind of get bundled together, and then they're just kind of in one little spot, held together. Okay? And so it's not a cell. It is a bunch of nerve endings, but instead of branched out, they're just bundled together, and so they're going to kind of act as a unit. But they're not an individual cell. Here we see a corpuscle, we see, yeah, always see our corpuscles. Here are free nerve endings. So it just kind of comes up and then just kind of branches. So in our somatic senses, we have a number of them. It's very tempting and it's kind of useful. It is touch. It's just then subdivisions of touch. And so there is touch. A touch is going to more or less just be, am I touching something? And then, yes, I'm touching it. What does it feel like? What is its surface? How rough is it? How smooth is it? It's not going to be anything about how hard am I pushing on it? How hard is it pushing on me? That is going to be pressure. Pressure is how hard. It's not going to tell you anything about what it feels like. All it's going to tell you is how hard is it pushing on your skin. And so that is going to be your Pacinian corpuscles in the skin. It's also going to be what's in your muscles. That's going to tell you whether your muscles are stretched. If you stretch yourself and you get to the end, and you can kind of feel that, right? If you're stretching and you go as far as you normally go, then you go a little bit further, you can feel tightness in your muscle. Or if you sit for a long time, then you stand up, that first couple steps, you feel tight. That tightness feeling is pressure. We also have some highly specific things, like vibration. Vibration is actually a different sensation than touch or pressure. Itching and tickle, those are definitely different. We have thermoreceptors that are going to tell us whether we're touching something hot or cold. We have pain receptors, which are also called nociceptors. And there are proprioceptive receptors, which tell us where our body, how our body is oriented. If I'm standing here, even without looking, I know that my feet are pointed forward. Or if I put my hands behind my back and I just kind of move them around randomly and I stop, I know that my left hand is above my right hand. That is based off these proprioceptive receptors. Yeah.
So why are some people ticklish and others aren't?
It has to do with how sensitive these free nerve endings are. And so, are there people who are not ticklish at all, like nowhere? Honestly, I don't know why. I would think it probably has to do, for you, it probably has more to do with how likely they are to get overstimulated. For other people, where they may be ticklish in one spot but not another is probably based on genetics, how many of these free nerve endings you have in each place. Imagine if the back of your arm. If you have 100 nerve endings, that may be enough to tell whether you touch it. But if you have 1,000 and you lightly touch it, then you have 1,000 sending a signal, and that's going to cause a tickle sensation. Then we get to special senses. Like I said, to me, these are the more interesting. The nose has a 10 to 100 million receptors for smell. And smell is an interesting one. It's special because it's the only one where there is a direct connection between your brain and the outside world. You have your brain, and then coming out of the brain is one of those cranial nerves. And then that cranial nerve literally comes into your nose and then puts little fingers down. And those fingers contact the outside world. And so it is possible to get from your nose to your brain. And people are looking at using that as a way to get things into your brain. It's hard to get drugs into your brain. We didn't talk about it, but there's something called the blood-brain barrier, and it's designed to prevent toxins and pathogens, bacteria and viruses from getting to your brain. But your body sees most of the drugs that we take as toxins, so it prevents them from getting in. So people are looking, if I can get a drug into that cranial nerve, it will travel up the cranial nerve into the brain and bypass the blood-brain barrier. For that question?
Is that how, like, people who do drugs, or anybody who snorts stuff? Because I know it messes.
I don't know for certain. I think that is more a way of.
Or is it just the lungs?
I think it's a way of getting into the lungs. Does anybody want to admit to know that? Okay. So, do I have a picture? Yeah. So we have our nose. So when we breathe in, the smell, we're actually smelling molecules. There are molecules that are floating around. Okay, we sniff that in. They're going to come up here into this big nasal cavity. And at the very top, we have a bone. And then on top of that bone is that cranial nerve. So that's this bright orange yellow thing coming through here. And then there are going to be, if you zoom in, little fingers that stick through holes. So there's a hole in that bone. And so you can see where the neurons here have dendrites that come down right up to that bone, and then the nerve actually kind of starts to come through the hole. And so, if you smell something, that smell can literally come right in and get up to that cranial nerve. Along that bone, right in this area here, is the olfactory epithelium. It's a layer of cells who serves multiple purposes to help us smell. So there are three types of those cells. The most obvious one are the olfactory receptors. And then there are a couple other ones that kind of serve the purpose of neuroglia. Remember, we have the neurons and then the neuroglia that help them function. And so there are supporting cells. The supporting cells are going to be things that maintain the environment around the receptors. And then there are basal cells. And the basal cells are going to be more like standard epithelial cells that are going to provide a platform. They're going to be more structural. They're going to create an epithelium, and then embedded in that epithelium are going to be the receptors and the supporting cells. Each of these receptors is able to respond to different scents. And so we have different receptors for sweet smells and sour smells and sulfur smells and things like that. When that molecule binds into that receptor, if I smell sulfur, the sulfur is literally going up into my nose, and that molecule is binding to one of these sulfur receptors. And when it does that, it creates a small impulse, makes it hit threshold. When it hits threshold, it's going to send an action potential up through that cranial nerve into my brain, so that my brain can integrate and say, I smell sulfur. So, looking at it a little bit more in depth. Remember, we have two nostrils. This is actually happening on each side of our nose. And so we have 20 holes, and those holes are in the cribriform plate. So hopefully you have a picture in your mind what the cribriform plate looked like. That was, if you took the top of the skull off, towards the front, there was a little oval shape, and then in that oval shape were a bunch of holes. The holes were called what? You remember? Cristagalli. We didn't talk about why they were there, but those holes are what these receptors are going to protrude down through. So the cranial nerve sits right on top of that plate, and its fingers stick through those holes. The axons of those receptors are going to come together to make that cranial nerve, which is the olfactory nerve. So the dendrites are what's going to stick through those holes. You have dendrites sticking through the holes. They're going to accept that signal, and then it's going to travel through the axon into the brain. The olfactory bulb, if you look at the olfactory nerve, at the very end, it gets kind of big. So it's kind of a bulb or a blob at the end of the nerve. You'll see that when we talk about the special senses in the lab. Once this whole process gets going, it's going to go up that cranial nerve, which is part of the olfactory tract, and then it's going to go to multiple parts of the brain. You don't need to know, though, which parts of the brain. We didn't even talk about the limbic system. So here we have the cribriform plate with the holes, and then down here we have the olfactory epithelium. And so in here there are the receptors. And so right here you can see where the end of the receptor is actually touching the outside world. And this is, so the outside world, you can see the little molecules flying by, and then that, what do you think is serous secretion? What would you normally call serous secretion? Snot. That's snot. Okay, so that is smell. Then next is taste. Taste, the official word or term for taste is gustation. And so if you see that term around in the book, online, or whatever, that is just talking about taste. And taste isn't nearly as complicated. There are 5 tastes. And what we interpret as a whole bunch or a whole wide range of tastes are just a combination of these 5. It's what are the ratios between those 5. That's going to give each individual thing that we eat or drink its own unique flavor. And so we have sour, sweet, bitter, salty, and then the weird one, umami. Okay? Especially in Western cultures, we don't use umami all that much. We don't add it to things. Do you know where umami really comes into play? Or how do we add it to some of our food? It's MSG. MSG that people are absolutely freaked out about. I mean, when I say MSG, do you have any sort of
Chinese food is where
Chinese?
it's from.
Yes, it's
Three
very
hours.
heavily used in Asian food. MSG got a bad rap a while ago for a couple reasons. Number one, some people are allergic to it. But if you're not allergic to it, you're not allergic to it, right? And also, people think it has a whole bunch of sodium in it. It has a lot of sodium in it, but it actually doesn't have as much sodium in it as salt does. And so, take MSG versus salt, they're kind of one and the same. It's not that big of a deal. But MSG is monosodium glutamate. It triggers that umami sense on our tongue. It's a very meaty flavor. And so if you add MSG to something, it makes it taste very meaty, even though it's not meaty. If you can imagine, if you've ever had lo mein, it's the very brown noodles, it tastes kind of beefy, meaty, but it's not beefy, it's not meaty. That's just MSG in there. Whereas smell only had one cranial nerve, taste actually has 3. Now some of these have other purposes also. Like the vagus nerve, we learned, was the one that goes down and controls our organs. The facial nerve also controls some of the muscles in our face. Okay, so these are not 3 specialized cranial nerves just for taste, but there are 3 cranial nerves that go down and touch our taste buds. And so depending on which of these flavors you're tasting, it's coming up through the different nerves. And a lot of these different tastes are located in different places in our mouth. Some are on the front of our tongue, some on the back, some on the side, some are actually on the back of our throat. And so those different nerves are going to attach in different places. Once those sensors taste something, they're going to send the signal to a number of places. There isn't one single place. There is a spot in the brain that's considered primary gustatory center. Remember, gustatory or gustation is just taste. So there is a major taste area in the brain, but that is not the only place these signals are going to go. One thing that people normally have misunderstand is that when you look at a tongue, the bumps on the tongue are not taste buds. Mind blown, right? Though the taste buds are actually microscopic. Those bumps on your tongue are called papilla. And they actually come in different shapes. We're not going to talk about different shapes, but they come in all these creepy looking shapes. If you knew what they look like, if you're interested, go home and look at the shapes of these papillas. There's like spikes, there's like little sea anemones. Did you also know it's pronounced anemone, not anemone? Wow. And so the taste buds, like I said, are microscopic, and they're on those bumps. There's a whole bunch of taste buds on each bump. And so, big tongue, you zoom in on the tongue, on the surface of the tongue, and you can see these different bumps here. They're different shapes. Then if you zoom in more, this is a taste bud. Here this is called, the top of the bud is a taste pore. It's an opening in the tongue where the food can get down in, and then these are just receptors. And it's very much like a lock and key mechanism. Each receptor has a lock. It recognizes molecules that are a certain shape. And if you eat something with molecules that fit into that lock, it's going to activate these receptors. And so if it fits into that lock, our brain says it must taste like such and such. And so that's why things that are not even closely related can taste the same.
I was about to say, is that why people say a lot of things taste like chicken?
Yeah. Yes, because there are things in chicken that are very common in other meats. And so it doesn't have a unique thing like beef or pork that give it a unique flavor. And so chicken is just kind of a generic meat flavor. It's also why artificial sweeteners taste sweet. Most artificial sweeteners on a molecular level are absolutely nothing like sugar. But they have one little part of the molecule that fits into the sugar lock. And so it sets off the same receptors. And so because it fit into the same little hole, our brain just knows something fit into that hole, it must have been sugar. And so we taste sweet, even though the molecule that we ate is not sugar. We can't get any calories out of it. So we get the sensation of sweet, but our body can't metabolize it. So it just passes through and there's no calories. Next is vision. And so there are a number of things related to our vision. It's not just the eye. Certainly the eye is the most important part. But vision also has accessory structures. We have our eyebrows. Now, what on earth is the actual function of our eyebrows?
Expressions. Isn't it to
At this
protect?
point, in this point in evolution, it's probably most likely expressions. It's more people reading facial expressions. But we're saying that eyebrows play a role in vision. What do you think the role of eyebrow, even if it's minor, plays in vision?
Blocks the sun.
It helps block the sun. If,
I'm pretty skeptical about that.
Well, your eyes are
Well, why do you
focused
think
on
it's
hyper.
like they were
yeah, so it's a little bit anti-glare, kind of like the eye black, but most of it is if you squint. If you're looking at something bright, you squint and you bring it down. Now, some of you may have very sparse eyebrows and they may not work very well. I already know when I get to be 50 or 60, I'm going to have these gigantic, bushy eyebrows, and they're going to work very well. Right? And so they will, in fact, help block some of that sun. It's also going to help prevent perspiration from getting down into your eyes. If you get sweat in your eyes, you know it's not fun. You can't see very well, right? Especially if you have sunscreen on your forehead that then sweats down in your eyes. You're like, oh no, right? You put your hands in and you say, oh no, and you scream, right?
no one wears sunscreen.
Don't even get. I can talk.
Wear
I actually
sunscreen.
I made sunscreen for years, and I'm an expert on cancer. I can talk about sunscreen for hours, but not now. What about eyelashes?
Dust. Catch debris.
Protect. Yeah.
Debris. It's going to help keep debris out of our eyes, even though half the time it seems like the eyelashes are the debris in our eyes.
Yes.
Right? And if you have long eyelashes, you put on glasses, and every time you wink,
Yes.
they
Marks up the glasses.
mark up the glasses, and you feel it, and it gets really annoying. What about eyelids? Now we're getting a little more obvious.
Keep them
Blink.
moist.
It keeps them moist, and so every time we blink, we take the liquid, the tears, and we move it over our eyes to keep them moist. If something flies at your face, what do your eyelids do?
Blink.
They close. What else?
Squint.
Helps us to squint. If we go into a very, very, very bright location, we go from someplace dark to very bright. What's your initial reaction? You close your eyes. Your pupils are going to close, but even quicker than that, you're going to close your eyes. Because if your pupils are wide open and you go outside, you're going to do damage to your eyes. And so while your pupils are closing, while your eye is getting used to brightness, you're going to close your eyes. Help prevent that too much light from getting in.
I have a question.
Yes?
So, like for me, when I would go from a dark place to like going outside,
Mhm.
I sneeze. Why?
Those are called sun sneezes. And no one knows exactly why. It's thought that you go out from inside to outside, your eyes are not used to that. And so you get so much signal going from your eyes to your brain in such a short little instant, your brain almost doesn't know what to do, and it just kind of goes into a seizure. And you No, I do it too. It's no one, if you can figure it out, there may be a Nobel Prize. for
No,
it.
my daughter's father and my stepfather retina, all of them,
Mhm.
all do it.
Yeah.
Like, they all do it.
Yeah, I read
Like,
it was
I don't know
genetic.
anyone else who does that.
I mean, it probably is. I mean, but what is genetic that causes one person to have that over response versus
It
another?
has to be genetic for all three of them.
Receptors, how many receptors you have?
It could be. I don't know. We have extrinsic eye muscles. Extrinsic. What do you think the word extrinsic?
Outside.
Outside. These are going to be the muscles around your eye that turn your eyeball. Okay? That's all they are. They're eye muscles, they're external to your eye. They turn your eyeball. That's going to be or as opposed to the intrinsic eye muscles that we'll talk about later, the muscles in your eye. Then we have a lacrimal apparatus. Has a much more common name. Any guesses? It's the whole tear system. Tear duct, your tear gland, that whole thing put together. Lacrimal is tear. It's called an apparatus because there are multiple parts. There's actually a gland in the corner, upper corner, that creates tears. They drain across your eye into the tear duct, which then drains down into your nasal cavity. So that whole thing together is the lacrimal apparatus. The eyeball has 3 layers to it. Okay? The outermost layer is the fibrous tunic. And depending on where you are on the eye, it can either be the sclera or the cornea. The sclera is the white of your eye. If you look at an eyeball and you are seeing white, you are seeing the sclera. That is the outermost layer of the eye. But part of the outermost layer of your eye is not white. Where? Right in the center, where you see through, right? That is the cornea. And so the fibrous tunic is the outermost part. It is rigid, it is tough. And it has a white part and a clear part. The white part is the sclera, the clear part right in front of your eye is the cornea. And so if you've ever known anybody who had a scratched cornea, they got something in their eye, they got a scratch on the clear part on the front of their eye.
Is that where they do the like LASIK eye surgery?
The LASIK eye surgery is below that, but to get to below that, they have to literally cut the cornea, peel it back, work under, and then put it back.
Take me away.
Underneath the fibrous tunic is the vascular tunic. And then this is made up of 3 parts, depending on where you are in the eye. And we'll see these. These aren't nearly as simple as white versus clear. There's a choroid, ciliary body, and the iris. You know what the iris is, right? What's the iris?
The color part.
It's the colored part of your eye. It's actually what's going to expand and contract to control how big your pupil is. So it's going to expand and contract to control how much light is going to get in. So that's going to be the part in front. Ciliary body is going to be nearby. And then the choroid is going to be in the back. It's underneath the white of your eye. And then in the very, very back of your eye, on the inside is the retina. So it is only going to be kind of in the back half of your eye, and the retina is what actually receives the light. Everything else on the way in is only for the purposes of controlling how much light and the focus. On the back of your eye is the retina, which is the projector screen. Everything else is the projector. So the retina is going to sense that light, and the retina is going to send the information to the brain. And the retina has 2 parts. There is a neural layer, which is where the photoreceptors are, and then there is the pigmented layer. And these aren't as important, but you are actually familiar with the pigmented layer of the retina. How do you think you are, I guarantee all of you, have seen the pigmented layer of your own retinas?
Color, right?
The
Close
color,
your eyes,
when,
it's the dots. No?
when you talk about blue eyes, green eyes, brown eyes, that's the iris. It's really, really noticeable in animals.
Oh, when, at nighttime, when
What about
you shine the
nighttime?
light and it's like yellow or whatever.
Yeah, so animals, you shine a light on their eyes, they get the bright green, right? Take a picture of somebody. What happens? With a flash. You get red eye. It's the light bouncing off the pigmented layer of the retina. And so, if we get to dissect an eye, you're going to see that. You're going to go into the eye, and in the back, where you see the retina, there's going to be a whole bunch of blue pigment.
It's gonna be animal eyeball.
It's sheep. If we do it, it'll be sheep. Well, technically, only animals have eyes. So, right? It can't be a plant eyeball.
He got you.
What?
Right, but it could be a human.
You said it's going to be an animal eyeball.
That's
And
it.
so I said only animals have eyeballs, because we are animals.
Specific.
How would that be?
Here, this is that lacrimal, the lacrimal apparatus. And so the lacrimal gland is your tear gland. And it is located on the upper outside for a reason. Why do you think it's in the upper outside corner?
So that it drains down.
Yeah. If it's on the upper side, gravity is going to pull it down over the eye, right? If you put it over here, and the tear duct is here, the tear duct has to be in the middle because it's going down into your nose. So if the gland was here, what do you think would happen?
Only one side of the eyeball.
Only one side of the eyeball would get tears. It would get made here and go right down the drain. Hmm? And so the gland is in one top left or top outside corner, so it will drain across. You don't need to know the individual parts of the tear duct.
Do
But
you know the flow of tears?
no. If you know that there is a tear duct, and then it flows from the tear duct down into the nose, that's it. Here's our eyeball. Okay? And so we've got let's just start from the middle. So the cornea is the clear part on the front. If we follow that cornea back, it becomes what? What is the outermost layer that's not cornea? Sclera. It's the white part of your eye. And so we have the cornea, and then the sclera there. That's on this side. So the cornea comes back, and now we have sclera. Underneath that, we have the iris, and so the pupil is the hole that allows certain amounts of light in. So it's going to get larger or smaller, and that is determined by the iris. The iris is either side of the pupil. The ciliary body is also part of the same layer. And so ciliary body and the iris are both part of the vascular tunic. The ciliary body is what's going to grab on to the lens of your eye and pull on that lens. The lens of your eye is basically a clear water balloon. And you can change the focus of the light going through that water balloon by pulling it. And if you pull it, it's going to get narrower. It's going to get wider, but it's going to get shallower. So the light's not going to go through as much of it, and where it goes through, it's going to be flatter. So if you have light going through a circular object, where it goes through is going to be very arced. So it's going to bend the light a lot. But if you pull it tight, it gets flat. So it doesn't go through nearly as much, and it's a flatter surface that it's going through. Yeah.
So when people have bad eyesight, like what's not working?
For most of us, it is your lens. Either the lens is cloudy, that is a cataract, okay? Or a lot of times it's not equally shaped. And so if it's oddly shaped, then it's going to not be able to focus far or close. In some situations, it's a matter of these muscles, the ciliary body, not working well. But for most times, that's a severe problem. Where you just get old, it's your lens becoming deformed. Yeah.
That was my question.
I have a question. So people that are like legally blind that can't see objects, but they know something's there, but they
Mhm.
can't read it. Would that be the same thing?
My guess is most people who are that bad, it's a retina problem.
Okay.
Or there's just something genetically, or trauma, if they had some sort of trauma to their eye, that would destroy something in this mechanism.
When they do eyeball surgery, do they suture the eyeball back up?
They fold it back over.
And it just like goes back together?
I think it actually does kind of hold itself together. Yeah. I don't know that for sure. But
You're not allowed to play any rough. You're not allowed to do anything rough afterwards.
yeah, I think imagine like saran wrap. It's basically what it is. And so saran wrap kind of holds itself down. And so they just flop it back down. And the outside layer of your eye heals very quickly. And so once you get just a little bit of healing, it'll hold it in place. And then it can continue.
I do this.
They do?
Ew.
I work in that industry.
Oh, okay. With a needle? Or
I don't know how they do it. I've never been in the operating room, but they have sutures.
I think my brother went into like a palmetto bush and it got his eyeball.
yeah?
So then he had stitches and his eyeball that automatically dissolved.
Yeah, like
And now it's like permanently dilated.
That's
what
crazy.
I like.
My brother's had LASIK. So
No, thank you.
Not me.
No.
Not me.
No.
who doesn't like lasers or blades in your
And then, in the back of the eye is the retina. Okay? The retina here is the yellow. And so it starts here, and goes back. The retina, as you go back, actually turns into the optic nerve. So this is the optic nerve that carries the light information to your brain. And so that nerve comes in, and really all it does is spread out. Makes a big, wide, flat surface, and that is your retina. And so, if you look into someone's eyes and you see their retina, you are seeing their brain. If we take the eyeball, we can break it down into two cavities. Okay? There is the anterior chamber and the vitreous chamber. The anterior cavity, the liquid there is aqueous humor. The vitreous chamber has vitreous fluid, or also called vitreous body. So if we come back up here, the vitreous chamber is everything behind the lens. That is the majority of the eyeball. Okay? And that vitreous fluid, the vitreous body back here is just kind of a medium for the light to go through. It serves no purpose other than to allow the light to go through something and to provide pressure inside your eyeball to keep your eyeball inflated. You ever heard of glaucoma?
Yeah.
Do you know what glaucoma is?
Nope.
It's increased eye pressure. It's that water balloon, too much pressure. And so if you have very high pressure, it's pushing on all of this stuff. It's pushing all this stuff forward, and more importantly, it's pushing on that retina. And the retina is alive. And if you push on that retina too hard for too long, it's going to kill the retina. That's what glaucoma does. And so a lot of times, to treat glaucoma, you take eye drops that decrease that pressure. And if we decrease the pressure, for a lot of people, the problem is then gone. At least temporarily. You have to continue taking eye drops. This is your pupil. Okay? We know when you go into the light, let's take a break. Take a break. We'll come back at, how many slides do we have? We'll come back at 1/4 on that clock, almost 15 minutes.
So you made your own sunscreen?
I made sunscreen as part in a company. I wasn't like Etsy selling sunscreen.
Name wasn't on it?
So is sunscreen like really a thing? My
Oh.
last teacher said sunscreen is bad for you, don't wear it. Because
Thought we could just put my skin off.
No, I actually, if we ever have time, I have a PowerPoint about sunscreen that we can go through.
What? Why don't you just post it on Canvas?
Because there's no words in it, it's just pictures.
Oh, he said he's got PowerPoint.
It's on sunscreen.
Are we talking about rods and
Yes.
I want food, so I'm like,
Is there a difference from like SPF like 50 to SPF 100? Like functional wise?
Yeah, I'll explain on this.
I've heard
Yeah.
crazy things.
We'll talk about rods and cones, but not too much.
I have a problem with mine. I have a lazy eye.
But lazy eye is muscle.
That's what they, I don't know, they said I have a problem with my
I mean,
rods.
lazy eye isn't a technical term. But I always thought lazy eye was an issue with the muscle or the nerve controlling muscle.
My dog has a lazy eye.
It only does it sometimes. Like if I look at him, I see two heads. Like I see two of him. Like when I look at you like that, can you see this eyeball looking up?
Yeah, up to
Yeah.
right.
I see double vision. I can control it. Like you're a single person.
Yeah.
When I'm really, really tired, I have double vision like all the time.
I'd be terrified to drive that machine.
Oh, and the lights when people have their like brakes on
Oh, I
coming at
hate
me,
that.
it messes me up even more. Like
Yeah.
my head is like turned like this, but I'm looking this way. That's the only way I'll see. It's like a single.
My night vision is bad enough as it is, without only seeing one of each thing.
I wear sunglasses sometimes to be able to see one road at nighttime.
Why is that lady wearing sunglasses at night?
Your sunscreen question.
Yes.
And so it's a very, very common thing for people to say there's no big difference between them, right? Because they say one blocks 99% of the light and one blocks 99.9% of the light. 0.1 or 0.9% difference. The problem is they don't understand math, okay? And so it's not about the amount of light that it blocks. It's about the amount of light that gets through. And so, if you have a sunscreen that blocks 99% of the light, how much light's getting through?
1%.
1% is getting through. If you have a sunscreen that's marginally better, and it blocks 99.9% of the light, how much light's getting through?
0.1.
0.1%. How much more light gets through with that one versus that one?
0.9.
In terms of a ratio.
A ratio?
How many times bigger is that than that?
100?
10.
Okay.
And so that sunscreen lets through 10 times as much UV as that one does. And so yes, you are at this point you are dealing with small amounts of light. But if you can limit the amount of UV that you are exposed to by a factor of 10, it's worth it, right? It's like if you buy a lottery ticket, you have a very low chance of winning. But if you buy 2 lottery tickets, you have double the chance of winning, right? But here, you don't want to win, because winning is confusing. And so if you can have a 10th as much light coming through, assuming a linear relationship, which it's not, but you would have a 10th of a chance of cancer.
So is that why the higher the SPF is, the thicker it is?
Yeah, and so if you look at the back, at the active ingredient label, the higher the SPF, generally is going to be more active ingredients, and of each of them that are in there, there will be more of them. More of it, you know what I mean?
Yeah.
Like 5 versus 3, and the 5 that are there, there's a lot of them.
Because that's interesting. I figured it didn't matter anyway. I was going for the high number.
If you go for a high number, yeah. once you get high enough, then it turns into another mathematical thing where,
Like SPF 100,
yeah.
for example.
So let's just take an example, SPF 100 versus SPF 120,
Okay.
okay? Let's say SPF 100 is 99.9. I'm just making numbers up here. Okay, let's say SPF 100 is 99.9. And SPF 120 is 99.91%. Okay? This is done in a scientific experiment. The SPF is literally, they put sunscreen on somebody's back and they shoot a UV gun at them and see how long it takes to burn, with and without sunscreen. Okay? So if a SPF 100 means on the spot of their back where they put the sunscreen, it took 100 times longer to burn than in the spot next to it where there was no sunscreen.
Oh, okay.
That's literally all it is. And so every time you do an experiment, there's always error involved. If you take a measurement, if I ask you to give you a ruler, tell you to take the measurement of this marker in millimeters, a very small unit, 100 times, you'll give me a whole bunch of different answers. There's always error involved. And so now you're dealing with such a small little measurement, you're in the error. And so you could take one sunscreen, test it once, and you get 100. Test the same thing again, get 120. But if you get a result saying 120, you can put 120 on that bottle.
So you could get a 100 and test it 100 times, and you can just put the highest number you get out of that.
There is a limit to the number of times you can do it. But if I'm a company, if I'm a generic company, okay, and I make a sunscreen, and I sell that sunscreen to Walgreens and CVS, so they then put their brand names on it, they each have to test it, okay? Let's say CVS, it's 100, but Walgreens comes back 120. Walgreens put 120 on their bottle, CVS has to put 100, but it's the same thing. Once you get the lower SPF, then it's more of a real difference, because now you're dealing with maybe this place or this place in the number, and so the error is much smaller than that. So then you know there are real differences. And so what I do, I look for a high one, and then I know what ingredients to look for. I say, okay, I got these 5 bottles. They all have high SPS. I'm going to assume they're all basically the same. Based on the ingredients, which one do I want?
So what ingredients do you look for?
It's like, how much time do we have on the break? So the saying, well, how does an aerosol sunscreen work compared to like a lotion?
Aerosol is a chemist's dream. It is incredibly simple to make. The lotion one is very difficult to make. The aerosol one, all it is you take active ingredients, you dissolve it in ethanol, and you put it in a pressurized bottle like off. That's all it is. Whereas the lotion one, if you have an old lotion, any sort of lotion, sunscreen or a skin lotion, what happens to it?
It gets nasty.
What do you mean? What do you mean by
It
nasty?
dries out.
It dries out. You can get the oil on top.
Yeah,
It's
it separates.
Right, it separates. Because now without going into how it's made, there's water and oil there, and it's mixed. And you have to mix it well enough that it stays mixed. But you can't do that permanently. But if you dissolve something in ethanol, it's dissolved in ethanol. I don't care what you do to it. A million years from now, it's going to be dissolved in ethanol. So it's much
She's like, you
easier.
want it? She's like, I have Mountain Dew.
The only problem with the ethanol one
I'm
is
like, okay.
if you spray it and then you don't rub,
Yeah.
you can get uneven coverage.
For free.
And so
We have here something.
I used to use the spray can because I was fishing. And so I would spray myself without having to get it on my hands, which would then get on what I was doing. And I would spray myself and I would spend all day out in the sun, and I'd get home and there'd be a little
Blotches.
bright red quarter-sized spot on my arm that I missed. And so you really then have to spray it and then rub it. But if you're going to spray it and rub it, you might as well have used the lotion to begin with, because the lotions are cheaper.
That spray crap is stupid.
And with the spray crack, you get it on your eyes, you spray yourself, and you actually inhale. And you're like,
It's horrible.
You waterproof your lungs for a week, basically.
It's horrible.
Yeah.
Anytime I do it, like, I always get, like, weird sunburns. Like,
That's what we were talking. If you don't spray and then rub,
Yeah.
you get the weird patch.
yeah, which is what's the point?
Aerosol.
What's the point of the spray?
And it's also harder to see where you have it. With the lotion,
For my
it's white
face, I
until it
do.
dries.
Especially stuff that's supposed to go on
And
dry.
I don't,
Yeah, it's you don't.
I do,
Is
because
it there or is it
I'm
not?
trying
And
to
how
die.
much is there? It's hard to tell.
I'll
from
either
sun,
burn or
from
burn.
skin cancer. My whole family, everybody has skin cancer, so I'm like, fucking sunburn, motherfuckers.
I spend a ton of time up in the sun and I'm pale. I'm
Same.
Yeah.
white. And so at this point, I cover up. I mean, I'm fishing in Florida afternoon, summer, sun.
And long sleeve and
And
stuff.
well, if it's really hot, I have a short sleeve fishing shirt, and then I have sun sleeves, so they're skin tight and a cool fabric, and then I have sun gloves. And so it's just the outermost phalange showing, right?
Educational now.
And then I wear a hat, sunglasses, and a buff. And so I put
Damn.
sunscreen on my temples.
To go
because-
You
fishing?
prepared
To
for
go
bees
fishing.
or something?
To go
So
fishing.
the only skin you see is around my temple where the sunglasses don't cover.
People gonna think you're trying to rob
The
them.
tips of my fingers, and then about that much skin on my leg because I wear shorts that are 4 sizes
You
too
wear
big.
high socks.
I wear socks, pull the socks up, and then I wear shorts that are 4 sizes too big, so they hang way down. And
Where
so
do you
there's
go
like that
fishing
much skin
at?
on my leg. What?
Where do you go fishing?
I have a boat, and so I go all over.
Go see the key one day. I gotta see this.
It's a fresh
I
water.
gotta
Yeah, I think we need a picture.
That's great.
He's like, I got one.
I got all kinds of pictures.
Proof or it didn't happen.
I really hope you're wearing camouflage so you just all come out the same.
We're like, what are you doing? You fishing or are you robbing?
Do you go to Mass Pro?
Columbia should have been like a suit.
Bass Pro
Don't
stuff.
they have, I've
They're
seen
overpriced.
stuff that says
I don't
that
really
shirts and
fish.
stuff
I just
like,
go there to look at
SPF.
Almost a fishing
SPF, I
life.
don't.
So
I was raised that way.
those
It's not
are
like you don't fish, it's just-
number one. They basically do the same exact thing as sunscreen. They just test how much longer it takes.
If you didn't get to know.
Those numbers are not real.
It's not, it's just a matter of coverage of material over your skin.
It's how densely woven it is. But those numbers are
I
not
don't trust
trustworthy.
it. Yeah, I don't.
Because they say like a t-shirt is like an SPF 4 or something, but most people will not burn through a t-shirt no matter how long you stay out.
Yeah.
And so
I do. I burn through t-shirts if I swim in and stuff. Because my mom always thought that was the greatest way to protect me from the sun, and I always burn through it. So, I don't know.
I saw on Shark Tank somewhere like SPF like swimsuits for kids. He said they're not real.
The
Yeah, just
SPF
blocks the amount of
shirts?
sun, that's all it is. There's actually
So-
no.
The SPF shirt works.
this
I
day
mean,
was a
she
little
never
bit
gets
cooler,
burned.
so I was actually wearing a long sleeve shirt. But you can
Oh my god, he really did.
so you can see a little bit of
Oh, that's awesome.
long sleeves, buff, hat, sunglasses.
You ready to go into beehive?
Where is that?
No, I can't tell you.
Uh-huh.
She's
Might
gonna get fishing gloves.
stalk him.
Oh, yeah. 'cause
We don't know.
I
We
would
know what
get
he wears
stuff.
now, so you know, we never know. Uh-oh, look at him.
Johnson Lake.
He's like, oh my god. Oh
No,
my
because
god.
they asked you if you had Facebook and you're like, I don't use Facebook. I was
It's
like,
the same
dang,
concept
we're
with,
not
like,
friends, we're just sit next
bugs,
to each other in
or is it
class.
repellent?
They used to add me on
The
Facebook.
bug repellent and the sunscreen
okay.
combined. So
No, like
Okay.
clothes.
Oh,
Oh,
okay.
no, they actually put chemicals in there
Huh?
that are supposedly supposed
What do
to
you use
smell
on
and deter the
Instagram and
bugs.
Snapchat and stuff?
I don't really post
So
on Instagram.
that
I post
wears
on
out over
Snapchat.
time because
I
the
feel
chemical
lame, like
washes
my
out.
posts compared to everyone else on like Instagram. I'm like, I look stupid. I don't even
My
post it.
Instagram is mostly my kid, so.
But, like I'm following people who are like gonna be.
Did you guys see my picture I posted? Like at 3:30 this morning? On Snapchat? Look how cute.
Rosa and Tony.
Yeah, I saw that one.
I didn't see it. I don't think I've really looked at my Snapchat.
The lady, like I saw like the eye doctor and she said I need like physical therapy glasses to like fix it. So maybe it's not even like has anything to do with it.
Yeah, I think it has more to do with muscles.
I believe you. But she said I don't need glasses.
Okay, so we're gonna finish talking about the eye. You know, you've heard of rods and cones. Do you know what rods and cones are?
Nope.
Yes.
The rods and cones are literally the receptors. They are cells on the retina that are going to sense the light. The light's going to come through your eye, project onto the back of your retina, and it's going to hit those cells. And then when they hit those cells, it's going to send an action potential into your brain.
Okay.
Rods and cones, though, serve different purposes. Rods are bright vision, and cones are dark vision.
Like nighttime vision?
Right, I think I got that back. Just a second. I want to make sure I told you right. Cones are color. Yeah, sorry, I was backwards. Yes. Cones are color. And they're good in daytime. You don't have nearly as many of the cones, so you need a lot of light in order to see the color. If you go outside when our class ends, it's dark out, unfortunately, right? So you're not going to see color nearly as well. Your cones don't work as well. But at night, in low light conditions, you have the rods, which you have a whole bunch of, that are good at detecting black and white.
Oh, so you did say it backwards.
Yes, I did say
Okay.
it backwards. I apologize.
It's
Yes.
okay.
Rods are for black and white in low light. Cones are color in bright sunlight.
Don't shrimp have like more rods and cones than we do?
I don't know.
What?
I have
Who
no
knows that?
idea.
It's like they have like a lot more than we do.
Rods are black and white, right?
Yes, rods are black and white.
So when you're colorblind, it's because of the cones.
It's a cone.
Really? So how come, or never mind. Never mind.
So the light rays are going to come into my eye. What's the first thing the light ray is going to hit when it gets to my eye? Cornea. Cornea is the clear outside part. The cornea also is going to help focus the light. Think about our microscopes. Remember, there are two focus knobs. There's the coarse adjustment, and then there's the fine adjustment. The cornea is sort of like the coarse adjustment. It gathers all this light coming from all these different angles and does a ballpark focusing of it to get it at least to the lens. Because if we have light coming from the very edge of my eyeball, it's not going to hit my lens. It's going to go right past it. So it's going to hit the cornea, and the cornea is going to bend it and send it to the back of my eyeball. Once it goes through the cornea, though, it's going to hit the lens, and the lens is going to do the fine focus. It's going to really allow us to see things clearly. If we're looking at something up close, the lens is going to become more curved. If we don't have proper curvature, we get bad eyes. And so if we are nearsighted, it's called myopia. If we're farsighted, it's hyperopia. And if it's just weirdly shaped, it's going to be called astigmatism. Yeah.
Okay, nearsighted is when you can see near but not far.
Yeah. So nearsighted is, yes, you can see near, but not far. And so this is a normal eye. And so the light is coming in, it's getting focused on a spot on the retina in the back. In a nearsighted eyeball, it's not focused correctly. And so the light gets focused on a spot kind of in front of your retina. And so it's like if this projector was focusing as if the screen was here. If it thinks the screen is here, but the screen's actually here, it's not going to be in focus. Because by the time it gets back here, this light ray is going to be down here, this one is going to be up here. And they're not going to converge like they're supposed to. If you have glasses, it bends the light so that after it then goes through your actual lens in your eye, it's correct. If you're farsighted, it's focusing on a place back behind your retina. And so you have a different shaped lens on your glasses that when you combine that lens with your eye lens, it now focuses on your retina. If you look at something close to you, you become cross-eyed, right? If you are able to purposely cross your eyes, generally, I think people look at the tip of their nose and make their eyes cross. That's called convergence. When you look at something close, your eyes undergo convergence and they move together. That's another reason why people can have trouble reading when they get older. If you're reading, you're looking at something nearby. That's another mechanism that has to happen for that to work. Your eyes have to be able to move in towards the center. So if those extrinsic eye muscles become weak, you can't move the eyes in as well, and so you have trouble seeing close things. And so you're able to overcome that then with reading glasses. Constriction of the pupil is, remember we talked about that as part of the autonomic nervous system. We can't control that. You can't go into a dark room and try really hard and keep your eyeballs, your pupils small. If you go outside into the sun, you can't purposely keep your pupils large. Yeah.
If you have astigmatism, can you either have like myopia or hypertrophy?
They're completely separate things.
Because I have astigmatism, but like I can't see far away, like I can see closer.
Yeah, and so you are nearsighted with astigmatism.
Oh, so I just have both.
Yeah, you can be nearsighted, you can have astigmatism, you can have both, you can have any combination, farsighted, astigmatism, whatever. It's just if you have astigmatism, your glasses and your contacts cost 5 times as much.
Yes.
Yes.
Yes, dude. That's why I am not wearing them.
Okay. So, like we talked about, rods are going to be more nighttime, black and white. Cones are going to be bright light and color. The rods and cones are the cells. They're actually cells. There are pigments in those cells that are going to absorb the light. In the rods, it's rhodopsin. That one's easy enough, right? Rhodopsin in rods. In cones, it's photopsin. Photo like light, but they're both light. If you want to remember light as in bright light, is cones, that may help you to memorize it. But you need to know the purpose of rods and cones, and then which pigment is in each. We're not going to talk too much detail about how this information is going to get to the brain, but it's going to go from these sensors, from these cells, through an optic nerve, into just behind the eye is something called the optic chiasm. It's kind of where the images from the two eyes come together, because we have two separate sets of rods and cones. The image is going to come together in the optic chiasm and then move up the optic tract to the thalamus. Once it gets to the thalamus, we said the thalamus was kind of the part of your brain that determines where in the cerebrum things go. Everything goes into the thalamus, and then the thalamus delivers it to the exact spot it needs to go. And so it's going to go to multiple places. There is a primary visual area that we'll see, and that is in the occipital lobe, which is where? Back. And so the primary visual area is in the back. And it's interesting. We have two eyeballs. If the primary visual area is in the back, it's in both hemispheres. Which side of the brain do you think the right eyeball attaches to?
I'm going to say the right.
If it
Everybody
attaches,
said left.
let's say we have the right eyeball attaching to the left. Okay? What side of the body does the left part of the brain control?
The right.
The right. And so if I am standing here and something comes flying at me from the right, which eye is going to see it first?
The right one.
The right one. which side of my body is going to have to react to that thing flying at me? My right. If it's coming from the right, it's going to hit the right
You want your
eye.
right to
And
react.
so it has to connect to the left side of the brain so that the left side of the brain can tell my right side of my body, stop it.
That's cool.
Oh, okay, that makes sense.
So here you can see it crossing over. The left eye goes to the right side of the brain, the right eye goes to the left side of the brain. You don't have to worry too much about that. But it goes from the eye to the thalamus, and the thalamus to the visual area. Then there's hearing. To me, hearing is my favorite. It's much more mechanical and straightforward. Things literally move, and that creates sound, that creates vibrations, which then get transferred to your brain. So when we look at an ear, we break it down into three parts. There's the external ear, the middle ear, and the internal ear. The external ear is going to be where you can safely stick a Q-tip. Okay? When you start feeling pain, that means stop. Okay? The box tells you don't even stick it down there at all, right? But nobody buys a Q-tip to not stick it in their ear, right? And so the external ear is going to be everything outside of your eardrum. If you stick that Q-tip in far enough, you're going to hit your eardrum. And if you do, you're going to know it. Okay? So everything outside the eardrum is the external ear. And its purpose is to just collect the sound and bring it in. The external ear is this part of your ear, but also the canal going into your head. The middle ear is going to be on the other side of the eardrum. There's going to be some levers and things in there that are going to take the vibrations of the drum and transfer them further in to the internal ear. The internal ear is an enclosed structure that has a whole bunch of very specialized receptors, both for hearing and also balance and equilibrium. So the external ear is the auricle. It can also be called the pinna. That's the floppy part of your ear. The whole thing. All the part outside of your head is the auricle. Then there's the external auditory canal. That's the hole going into your head on the outside of the eardrum. And then the eardrum itself is considered part of the external ear. The middle ear has the auditory tube, which is usually called the eustachian tube. Some auditory ossicles, which are just going to be levers. They're simply levers that are going to move. And then there's something called the oval window, and the oval window is going to be where the levers connect to the internal ear. So here is the external ear. You have the auricle, and then the canal, and then the eardrum. Between the eardrum and this snail-looking thing is the middle ear. And so these are these ossicles. We'll talk about how they work, but there's three of them, called the malleus, the incus, and the stapes. Literally all they do is when that eardrum moves, it causes these to move, and they're connected to that, and so it pushes and pulls. Every time that eardrum moves, it pushes and pulls on this little connection. And then this is the eustachian tube. Where do you know eustachian tube from?
They put tubes in your ears.
Tubes in your ears are actually little tubes in your eardrum. Do you know why they put tubes in your? I had tubes when I was little. Do you know why they did that?
The fluid. It's
Right.
Ear.
If you get fluid back here, it's a great breeding ground for bacteria. And so you get ear infections. Most of us, though, we're adults, majority of us don't continuously get ear infections. That's very common for kids. When we get older, the eustachian tube works better. This pinches shut and opens. If you fly in a plane, you get the pressure build up in your ears, you pop your ears. What happens is this opens up and it allows the pressure to equalize. This goes to your nose, which connects to your throat, which is connected to the outside. Yeah. And so if this pinches shut, pressure outside changes, but the pressure in here can't equalize. So as you go up in the atmosphere, pressure here goes down, and so the pressure inside pushes on all of this stuff, and it gets very painful. But if you hold your nose and blow, which every doctor tell you not to do, it creates enough pressure here that it quickly pops that open for instant and lets everything equalize. What's supposed to happen is every time you swallow, that opens up. But if you have a cold or something and you have some inflammation there, it's swollen shut. And so when you swallow, it doesn't open like it's supposed to. We have external ear, middle ear, and then there is the internal ear. And it has a number of parts. This is where the magic really happens. There's something called the bony labyrinth and the membranous labyrinth. So the bony labyrinth, you can imagine this snail is embedded in bone. Okay? That snail, though, is flesh. It is living cells. And so in order for all of this to go through the bone, there has to be canals in the bone. So wherever these loops are, if that person died and this all decomposed, there would be little channels going through the bone. So those channels are the bony labyrinth. The membranous labyrinth is going to be the living stuff inside of that. So these kind of hoses, the tubes going through that. Yeah. And then there is the cochlea and the spiral organ, which is also called the organ of Corti. The cochlea and the spiral organ are what's going to actually sense our hearing, particularly the spiral organ or the organ of Corti. So coming over here, this is the cochlea. This whole thing here, not these little loops though. Everything below that loop. And so you can kind of imagine, you see it wrapping in a spiral. You see that? It's a channel that wraps around. And so the sound vibrations are going to go through here, and when they get to the center there, they actually hit the spiral organ or the organ of Corti. So the cochlea is that physical spiraling, and then the organ of Corti is the sensor at the end of the channel. These things up here, these semicircular canals, are what's going to help us determine balance and posture. Here we can see this better. And so you have this part up here, which is going to be for balance, and then this part is for hearing. So this is that, and this is that part there. So if we hear things, the auricle, the outside part of our ear, is going to take that sound, put it into our external auditory canal. It's going to hit that eardrum, and the eardrum is going to vibrate back and forth. When that eardrum vibrates back and forth, it moves those bones, those levers in the middle ear. And when they move, they're going to transfer that all the way to the oval window, which is where they connect to the cochlea. When that oval window gets moved in and out, it transfers vibrations into that cochlea, that spiral snail-looking thing. It then moves through that all the way to the end to the spiral organ, the organ of Corti, which is connected to a cranial nerve. So vibrations outside, eardrum, the eardrum transfers it to the cochlea, it spirals through the cochlea to the end of the line, and then the spiral organ transfers the information to a cranial nerve. The other parts of the ear control balance and equilibrium. Okay? We have two types of balance and equilibrium. There's static equilibrium, and then there's dynamic equilibrium. Static equilibrium is going to tell me how I am oriented. if I closed my eyes and somebody came up here, picked me up, and tilted me on my side, I would know that, right? I would know that I was laying on my side. Or if you wake up in the middle of the night, before you even know what's going on, you know you're laying down. You just know it, based on how you're oriented. That is static equilibrium, and that's determined by the utricle and the sacule, okay? And the way this works, it's kind of, I wish I would have thought of this before. I always forget to bring something for an example. Imagine you have a bowl, okay? Just a cereal bowl, and then there's a marble sitting in there. If the bowl is flat, where is the marble going to be? Right in the center. If you tilt the bowl like this, where's the marble going to roll? Towards me. If I tilt it to the side, which way is it going to roll?
To the side.
It's going to roll to the side. And so literally in your ear, you have these little granules that sit there in these little cups, and as you tilt your head, the granules move in the cup. And so based on where the granule is in the cup, you know how your head is tilted. That's different, though, than dynamic equilibrium. Dynamic equilibrium is speeding up and slowing down. Think about when you hit the brakes, you know it. Even if you were riding in the car with your eyes closed and somebody hit the brakes, you would know it. If they hit the gas pedal and you accelerate, you would know it. If they turn, you would know it. We call those G-forces, right? The way this works is you essentially have, that's those semicircular canals, those curved channels. When I turn, there's fluid in those channels. And because of inertia in physics, the channels have to move with me. They're connected to me. But if I have a tube with water in it and I quickly move that tube, the water kind of lags behind a little bit. And so we can actually sense that water lagging behind. And if the water seems like it's too far to the right, that means the tube actually moved to the left. And so if you get the water whooshing around through those tubes, and if you go back, how many tubes do we have? Three. How many dimensions are there in space? 3. Up, down, left, right, forward, and back. So we have one channel for each dimension, X, Y, and Z. Any direction we move, it creates a combination of sensations in those 3, and we know what direction we're moving. That's just kind of showing what we talked about. There's way more detail there than you need to know. And that is the end. Questions?